All right, now we're going to cover chapter 7. I'm going to skip the intro part. Okay, that's all like how neurons work and stuff like that. That if you don't really understand, go back. There's a video I recorded for my anatomy class that basically explains all this. I included a link already on the bottom, okay, of the sheet that you guys received. So I'm gonna skip forward to the more important parts, okay? All right. So, okay, first things first, neurons have a resting potential of -70 millivolts, okay? And that's established by the electrochemical gradient, okay? So we have a sodium potassium pump, pumping stuff to the outside, right? It pumps all the sodium to the outside, all the potassium to the inside. And it generates 3 potassium for 2 coming in. So 3 plus charges leaving, 2 coming in. There's a negative gradient already there. There's also large negative molecules inside the cell that can't cross the barrier, like DNA and stuff, okay? That generates a negative potential inside the cell. Okay, at rest, there's a high concentration of K plus inside the cell and a high concentration of Na plus outside the cell. Okay, these two things are going to become really important in a little bit. Okay, so I can alter the membrane potential. Okay, this is called excitability or irritability. I can do it by opening channels to certain ions, okay? So if I open a sodium channel, for example, sodium will start rushing into the cell, okay? So positive charges will come rushing into the cell, and that would depolarize the cell a little bit, okay? Make it less polar, make it closer to 0 than at -70, okay? The other thing I can do is I can open a potassium channel and have potassium leave the cell, okay? And all of a sudden, I have more negative charges than usual, and so now I'm hyperpolarized. So I drop below -70 to -85 or something like that, okay? Okay, so a cell is polarized, right? So the charges are equal. If they were 0, they would be non-polarized, but they're not 0. In fact, they're very rarely 0. Okay, the membrane potential inside the cell increases. This is called depolarization. Okay, hyperpolarizations become more negative. So I can tinker with this anytime I want. Okay. So here's my resting membrane potential, RMP. I can depolarize it, okay? That would be a stimulation. Okay, here's my axon, okay? If I want my axon to fire, then I'm going to depolarize it. Okay, if I don't want my axon to fire, I'm going to hyperpolarize it, okay? The electrodes are in the wrong place here. They should be at the cell, or the axon hillock to be more exact. Okay. Okay. So depolarization occurs when positive ions enter the cell. So I explained that sodium coming in usually. Okay. Hyperpolarization occurs when positive charges leave the cell or negative charges enter the cell. Like the K plus example I gave you. The other example is chloride ions coming in. Okay, more negative charges coming in means the cell is more negative now. Okay, depolarization of the cell is excitatory, and hyperpolarization is inhibitory. You don't know why yet. I'm going to explain that in a couple of slides. Okay. Changes in membrane potential are controlled by changes in flow of ions through channels. Okay, potassium has two types of channels. The leakage channels, they're just called leakage channels, or the voltage gated channels. Voltage gated means it has to cross a threshold to flip. Otherwise, it's closed. Okay? Sodium only has voltage gated channels that are closed at rest. Okay? Okay. The voltage gated sodium channels open, okay, if the membrane potential depolarizes to -55. This is a threshold, okay? Above the threshold, you open up all the sodium channels. Below the threshold, nothing happens, okay? So every time I depolarize, I come closer to the threshold, okay? One depolarization is not gonna do it, okay? But every time I depolarize the neuron a little bit, I come closer and closer and closer to the threshold. Once I get to the threshold, bang, the sodium gets open and all hell breaks loose, okay? So, not all hell, but the membrane, it flips open the sodium channels. Sodium channels, once they open, all the sodiums will come rushing into the cell. Remember, all the sodium's on the outside of the cell, dying to get in. So once I flip that gate open, all the plus charges will come rushing into the cell, and then it will become actually positively charged, okay? Okay. So the sodium comes rushing in once you cross the threshold, okay? The membrane potential climbs toward the sodium equilibrium potential, and these channels are deactivated at 30 millivolts, okay? Okay, so the way I prevent these sodium channels from opening is I hyperpolarize the cell, okay? If I hyperpolarize, then I'm never going to get to 55, right? Anytime I want to decrease the likelihood of the neuron firing, I lower the membrane potential to below 70, okay? Okay, so here, okay? This channel, okay, is open, okay? What actually happens, really, there's a little ball. This is actually a really cool thing. There's a little ball here. That ball, after it opens, that ball will eventually close, and this, even though the channel's open, doesn't matter what the voltage is, that ball is stuck there, and it doesn't come off for a while. So while that ball is there, it doesn't matter what the voltage is, that gate is still closed, okay? That period where it's closed is called the refractory period, okay? This is a physical, like, it's literally a block, so there's nothing that can get through, and it doesn't matter what the voltage is, okay? All right, at about 30 millivolts, the voltage gated potassium channels open, and now potassium goes the other way. Potassium rushes out of the cell, and that makes it all of a sudden very negative. Okay? All right, so here we go. Okay, so I have a bunch of stimuluses. Okay, we'll explain what the stimulus is in a little bit. Okay, but this is magic for now. So the stimulus triggers this potential, so -55. Once it triggers -55, all the sodium channels open. And then all the sodium channels open, the membrane potential shoots all the way up to +30 before they before that little ball locks in and they close. Okay, this triggers, okay, the potassium channels, sorry, to open. Now the membrane potential goes the opposite way. In fact, it actually drops below the original 70. Okay, and then these channels close and it resets. Okay. Okay. So hopefully these make sense to you guys. Okay. So here is what happens to the membrane potential. Here is the distribution. Okay. Okay, so after hyperpolarization, repolarization actually overshoots the membrane potential and shoots it down to negative 85. Okay, this does not reach potassium equilibrium because voltage gated potassium channels are inactivated as the membrane potential falls. Okay, and then you have the sodium potassium pump resetting this up again. Okay, the sodium potassium pump wastes a lot of energy doing this. It's not cheap. Okay, and every time it fires, the neurons has to do this. This is why neurons are so metabolically expensive. Okay, because you have to do this over and over and over again, and then you have to build a bunch of proteins to do this. So it's neurons are expensive pieces of material. Okay. Okay. So this is the all or none response. Okay. You either the way I explain it in anatomy, which is more simple, is that neurons have one choice. You either fire or you don't fire, and that's it. All or none. Okay. Okay. Once you fire, that's it. That whole pathway, that you can't stop that. Okay. Okay. So the size of the stimulus will not affect the size of the action potential. It'll always be 30 millivolts. Blah blah. The size of the stimulus will not affect the action potential duration. Like you just once you open that gate, they go through. Okay. So no matter what the trigger is, the end result is all the same. Okay. Okay. Coding for stimulus intensity. A stronger stimulus will fire not harder but faster. Okay. A stronger stimulus will also recruit more neurons in a nerve to fire, and that's called recruitment. So we'll play that in a little bit. Okay. All right. So coding for stimulus intensity is the sheer number of action potentials, not how high the action potentials are. Okay. All right, the refractory periods. Okay. Action potentials can only increase in frequency to a certain point. There is a refractory period after an action potential when a neuron cannot become excited again. This is an absolute refractory period that occurs during the action potential. So the sodium channels are inactive, not just closed. So remember that little ball? That little ball makes it inactive, not just closed, but inactive. Okay, the refractory period is when K plus channels are still open. Only a really strong stimulus can overcome this. So okay, so here. Okay. Okay, so here It's the absolute refractory period where you can't do anything. Here's you need a huge plus. You need a huge sodium input to compensate for the calcium or potassium output. Okay. 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 So the chemical properties of neurons. So here's the threshold, it weakens over time. Okay. In theory. Okay. Okay. So what actually happens? Okay, is you generate an action potential right here. This triggers an action potential right there. The red part is in the refractory period. Okay, so you can't go backwards. You can only go forward. If you look at the blue right here, and this is the important figure right here. This should be able to send action potentials both ways. But because the red is in a refractory period, it can't fire that way. So we can only go into the yellow. So we can only move forward and not backwards, okay? This one right here can only move forward this way, but not backwards this way. That way, the action potential will only go down the axon. It's a one-way ticket all the way down the axon. Okay. Okay. The action potential in an unmyelinated neuron is very slow because you literally have to go every single step of the way. Okay. The reason why the nodes of Ranvier are important, okay, are because the signal jumps down the axon and makes it faster, okay? Saltatory conduction makes it faster. So this has just enough to trigger this. This has just enough to the change in action potential, just enough to hyperpolarize, just enough to depolarize this region right here. So it fires off action potential, which has just enough to depolarize this thing right here. So it has to have an action potential. Okay. So again, this part is red because it can't go backwards. Okay. Okay. Action potential is increased by the diameter of the neuron. This decreases resistance. Big copper tubes transmit electricity faster than small copper tubes. The myelination. Okay. So this is why, like, we test axons on squid axons, that they're big, huge things. Okay. All right. Okay. Introduction to the synapse. The synapse is a functional connection between a neuron and the cell signaling. In the CNS, this will be another neuron. In the PNS, it will either be a muscle or a gland. Okay. And it's either called a neuromuscular junction for muscle. Okay. This neuroglandular thing, what, don't worry about that. Okay. Blah, blah, blah. If one neuron is signaling another neuron, the first is the presynaptic neuron. So synapse, presynaptic, postsynaptic. Okay. Okay. The presynaptic neuron can send over the dendrite, the cell body, or the axon of the second neuron. So axonodendric and axonosomatic and axonoexonic synapses. Okay. Most synapses are axonodendritic, which is why you have so many dendrites. The reason why you have so many dendrites and that greatly increases the surface area of the axon, or sorry, neuron, and you can receive signals more in one place. Okay. The synapses can be electrical or chemical. They're mostly chemical. There's some that are electrical. Okay. The electrical synapses are just electrical signaling by gap junctions. And gap junctions, if you recall, are junctions of the cell where they're attached to one another and signals can travel straight through. Okay. Okay. So here are the gap junctions, and here you can see that the signal travels straight through. Okay. Most synapses are chemical synapses, and chemical synapses always release a chemical called a neurotransmitter. Okay. Okay. The synaptic cleft. So there we go. Blah blah. Okay. So here's the axon terminal. Okay. Here's the cleft. Okay. I mean, smaller than mitochondria, so you gotta realize how small that is. Okay. All right, the neurotransmitter is enclosed in a synaptic vesicle on the axon terminal. Okay, so the signal will travel down the axon. At the end of the axon, and the terminal boson or axon terminal, that voltage change opens, instead of causing another signal to fire, it opens up a voltage gated calcium channel. A voltage gated calcium channel opens, and that allows calcium to flood in. Okay? The calcium flooding in causes the synaptic vesicles to actually bind and fuse to the plasma membrane. And when they fuse, they open up, and that means their neurotransmitter comes right across the synaptic cleft. Okay? The greater frequency of action potential will increase in more stimulation of the postsynaptic neuron. So every time, like I have to do this time after time after time, every action potential only causes one signal to go across the synapse. I have to destroy that signal, and then I have to generate another action potential. Okay? Okay. Okay, so let's talk about this. Okay, so here, the action potential reaches the terminal axon. You open up calcium channels here. The calcium binds to this synaptotangent complex, which then causes exocytosis to happen, and you release the neurotransmitter. Okay, once the neurotransmitter is released, it's usually destroyed right away, and so you have to do this over again. Okay. Okay. So okay. The greater potential. Okay. When the ligand-gated ion channels open, the membrane channels change potential depending on which channel is open. Okay. I can open a sodium channel or a calcium channel. Okay. And that results in an excited, excitatory postsynaptic potential. Okay. EPSP. Okay. When I open up a sodium channel, positive charges come running into the cell and brings it closer to negative 55. So I'm exciting the neuron. Okay, so that is a EPSP. Okay, if I open, positive charges come rushing in, the membrane potential gets closer and closer to negative 55. If I open a K plus channel, plus charges come rushing out, or chloride channels come rushing in. Either way, the cell becomes more negative, and that's an inhibitory thing. So I have an inhibitory postsynaptic potential, or IPSP. Okay, a neuron is going to receive multiple combinations of IPSPs and EPSPs, and just determines which ones win out. Okay, if I have more EPSPs, then I'm likely to fire. If I have more IPSPs, okay, it's less likely to go fire. And also depends on how close they are to the actual axon, the axon hillock. Okay, so IPSPs and EPSPs are graded, which means I have to add up everything. Like your exams are graded, so I have to add up, all the correct answers and subtract all the wrong answers and stuff, right? So EPSPs and IPSPs are graded potential. Okay, at the axon hillock. Okay, I mentioned this in the anatomy talk. The axon hillock is a very important piece. Okay, I sum up all the EPSPs and IPSPs at the axon hillock. I hit negative 55, bam, action potential, that's the axon. I don't hit negative 55, nothing. Okay. Okay. So there you go. Okay. So I sum up, I sum up all my EPSPs and IPSPs, okay, here at the axon hillock. And then if I get opening, I send an action potential. Okay. All right. I'm gonna stop right here. So